So today we will be discussing about viroids, right? So what are actually viroids? Viroids are the smallest pathogen, are small pathogen. Now what is pathogen? Pathogen are the organisms which cause disease, right? So these are the small pathogen. So now who discovered it? What is this? So let's talk about this, right? So basically it was discovered by T.O. Diner. Guys, let me tell you a story in which you can remember the discovery and the name of a scientist in a very interesting way, right? So consider like this is my own fiction story. Don't consider this as a truth, right? So Diner, like was the scientist was the name of the scientist was Diner, right? So Diner went to a hotel for dining, like to eat food. He went to dining, right? Dining means to eat food, right? So he went there and he ordered potato. He ordered potato. But the when his dish arrived, when his meal arrived, he saw that the potato was quite elongated. The potato was quite elongated. It was not in the proper structure. So he was wondering that why this potato is not in the proper structure. Like what's the reason behind? In commonly usually potato are round or oval, right? But why these potato are not in good shape? Why they are elongated, right? He was wondering about this. So what he did, he researched on this potato and found that this potato is infected by a disease known as potato spindle tuber disease. He got to know that this potato is infected by potato spindle tuber disease. So, and he found that this disease is caused by viroid. So the infecting agent of this potato spindle tuber disease was viroid. Now you can easily understand that T.O. Diner, like the name of the scientist who discovered the viroid, is just a fiction story. Went to a dining in which he ordered potato. He saw that why these potatoes are elongated in shape, why they are not in that common shape which are usually present in the markets, right? Then he researched about that potato and he found that he came to a result that it is infected by potato spindle tuber disease. And then he found that the infected organism, like the organism which is responsible for this disease, is viroid. So in this way I guess you can easily remember the viroid, right? So now what is viroid? Viroid is actually single stranded RNA. It is single stranded RNA, but it has, like in some regions it has double strand too. Like in some regions double stranded RNA too. Like in it basically it is single stranded, but in some regions it is double stranded RNA too, right? So now actually, and the most interesting thing, like we know that in virus, what were the two main components of virus, right? First was the capsid, right? And other one was its genome, RNA or DNA, right? So basically this is capsid, right? And this is RNA or DNA, like both can't be present. So this was virus, right? This is virus. Now what about the structure of viroid? So viroid is basically a naked RNA. It's a naked RNA. It doesn't have the capsid protein. It is naked. It is naked, right? So this virus causes a lot of diseases, and when this, like the viroid, like, it causes disease in plants, but when this viroid enters the human, it causes hepatitis D. Hepatitis D. So basically, the hepatitis D, the hepatitis D is viroid, right? So basically now, like from now, 33 species of viroids have been discovered, right? 33 species have been discovered. 33 species have been discovered, right, of this viroid. So now let's discuss that how this viroid is infecting, like how it's entering, how what's the mode of transmission. So basically, consider this as a viroid. So, viroid is not like this. Viroid is this. So this is viroid, right? And consider like this is a cell. This is a cell, right? This is a plant cell, right? So what happens, basically here we are having the nucleus, or here we are having chloroplast, like it's a plant cell, right? Like this is the chloroplast, right? And in the center we are having this nucleus, right? So let me label this. This is chloroplast, and this is nucleus, right? So now what happens, actually these both have enzymes, right? They both have enzymes. What kind of enzymes? Like they have RNA polymerase, they have DNA polymerase, like RNA polymerase, and they have DNA polymerase too, right? So what happens, actually, this is the viroid, right? And this enters the cell through the channels known as, this viroid enters the cell through the channel known as plasmodesmata. The viroid enters through the channel known as plasmodesmata. So now what is plasmodesmata? Basically, plasmodesmata, you can say that these are the channels. These are the channels of the cell wall, or you can see the openings, or you can say the spores. These are plasmodesmata are the pores of cell wall. These are the pores of cell wall through which something enters in the cell wall, in the cell, right? Now through this plasmodesmata, basically this region is known as plasmodesmata. Here we also having, like, these are the small pores or slits in the cell wall, by which something can enter into the cell, right? So this by this plasmodesmata, viroid will enter, and it will go either in the chloroplast, or in the nucleus. Like these both contain enzymes like RNA polymerase, DNA polymerase, as I discussed Okay, now what will happen, this viroid, like when it enters the chloroplast and nucleus, it will, like by utilizing the enzymes known as RNA polymerase, like when it will, like it will utilize the enzyme RNA polymerase. RNA polymerase. This enzyme, like this viroid, will enter the nucleus and the chloroplast, and it will utilize the enzyme known as RNA polymerase. Now, by utilizing the RNA polymerase, it will make its copies. It will make its copies. It will make its copies. So these are basically the copies which are made by the RNA polymerase. Now what will happen when these copies will come in the cytoplasm? They will use another enzyme known as ligase. They will use another enzyme known as ligase. Now what is the function of ligase basically? The function of ligase is to join the nucleotides. Join the nucleotide. So basically viroids are like, these are stranded DNA, stranded RNA too, but they are in circular form. They are in compactly form in circular. They are in circular form. They are in circular form, right? So what will happen, this basically, this ligase will convert these stranded, like this stranded RNA into the circular ones. It will convert this into the circular. Can I remove this one basically? I hope you have noted this down, right? So what will happen, this circular form will be formed. Now these are the viroids and these are in the circular form, which are formed from the ligase. Like ligase is an enzyme which joins the nucleotides and it converts the stranded RNA into the circular form. And now this, like my cell has ended, but in basically this this viroid will move from plasmodesmata. It will move from plasmodesmata and it will go in the other parts of the, it will go in the other parts of plants. Now it will go there and infect them. How they will infect it, like, okay, number first symptom, like what it will do is, it will, like in the plant, it will move all around the plant. It will first go to the pollen grains. It will go to pollen. It will go to the ovule. It will insert itself in the pollen ovule. So when this pollen ovule will combine together and fertilize, they form a seed, right? They form a seed, and now this seed also contains the viroid, right? They are infected by viroid. Like viroid has been inserted into them. Now and seed also contains this viroid, right? And when this seed will grow, like when we will plant this seed, when we will plant this seed and it will grow into a plant, right? It will grow into plant, and now this plant is infected by viroid, right? How? But because in the parental plant, when the pollen and ovule are forming, they were fertilizing, the viroid entered into them. That's why the plant formed will also be infected by the viroid, right? Now what are the basically symptoms? Symptoms are that there will be the stunt growth, like the growth will be stopped. There will be stunt growth. Moreover, there will be, like, it will lead to stem necrosis and then death of the plant. And then we are having RNA silencing. RNA silencing. What happens in this? Basically, RNA silencing, it is a process in which the viroids silence the RNA of the plant. What happens actually? Consider like there is a stress condition. There is a stress condition in plant means that the temperature is high. It means this. So when the temperature will high, like how the enzymes will work, they will obviously denature, right? They will denature. But what happens are, like, there's a mechanism in the plant that they will develop heat shock proteins. They will develop heat shock proteins. Now what will happen, now this heat shock proteins will get attached to the enzymes, and now they allow, now they can easily survive in the hot condition, like now they can carry. But what happens in RNA silencing, the RNA, like this viroid does not allow RNA to produce heat shock proteins. It will not allow the RNA to produce heat shock proteins. What will happen? This will silence the work of the RNA of the plant. It will silence, like the many other work will be also stopped. So basically RNA silencing will occur. The virus will not let the viroid will not let to work properly. The mechanism of plant work properly. What other things can happen is that there will be chlorosis too. Now what is chlorosis? See, chlorosis is a disease in which, like, the leaf turns yellow. The leaf turns basically yellow. Why? Because there is, like, maybe the amount of chlorophyll has become less. Maybe the amount of chlorophyll has been decreased, so that's why the leaf will turn into yellow. And this disease is known as chlorosis, and it is also caused by the viroids. So I guess this basic viroid is clear to you. So let me show you the diagram of potato spindle tuber disease. Right? So you can see here, like, when the diner was doing dinner, he saw that why these potatoes are elongated. This is elongated. You can see that this is not the clear size. Yeah, this can happen, this can occur, but how this and this will occur? So obviously these are not the proper size, the proper structure of the potato. So that's why he wondered and he researched on the potato spindle tuber disease, right? So I hope this concept of viroid is clear to you. If you're having any kind of questions, you can comment me below the video, and I will be very happy, I'll be very glad to answer you guys.